Okay, so with no further ado, good evening, good morning, and welcome, and good night as well, and good afternoon. Welcome along to In Conservation With. My name is David Lindo. I'm really happy to have Per Alström, who's one of the dons when it comes to taxonomy. I also need to mention the fact that this whole series is being sponsored by Leica Sport Optics alongside King's Place, which is a music foundation in North London, and they basically are sponsoring it as part of their Nature Unwrapped season. So, Per, good evening and welcome along.
Thank you so much, David.
Good. And for those who weren't around the first time Per was in Conservation With, he has the distinction and still holds that distinction of being the first person I've ever interviewed on this who was actually suffering from COVID-19 at the time. The man's still standing. How are you feeling?
I'm fine, actually. Yeah, no worries at all.
And he's also a man of few words when it comes to some of his ailments. He just gets on with it. At the time, I remember you said the same thing, but I think you were a little bit tired, actually, because you did tell me afterwards that you felt a little bit under the weather. But I'm glad to know that you're feeling good.
I was extremely lucky, really. So it was very mild. But anyway, it wasn't pleasant.
Good. Now, have you been doing much birding since I last saw you? Because I don't know what the situation is like in Sweden when it comes to getting out and about with the pandemic at the moment.
Well, birding's never been a problem in Sweden, really. As long as you're outdoors, that's no difficulty. So since I've not been able to travel at all this year, I've been birding a lot more in Sweden than ever before. Well, not ever before, but for a few decades. So it's been quite nice, actually. I've seen quite a few good birds. Yeah, it's been mainly just local. I've just been walking around close to my house, and Uppsala University has actually just published 3 different films where I'm promoting suburban birding with some of my own video of things like Slavonian grebes and red-necked grebes breeding just behind my house, and it's been pretty nice, actually. It's not too bad.
Yeah, I've seen some of your films, and I must say there's the shots of kingfisher as well on top of the Slavonian grebes. Very beautiful shots. Now, just to inform you Zoomers as to who Per is, in case you are, not familiar, he is a professor of ornithology working at Uppsala University in the Department of Ecology and Genetics. He is a keen birder. You've been birding ever since his early childhood in southwest Sweden. But I remember when he first came on, I asked, was there a spark moment, as my American friends often ask? Was there a spark moment? And you said to me no, and you actually said that initially you were interested in bears.
Still am. Still like bears, yeah.
Yeah. So
actually, since we last met, I had the pleasure of having beautiful views of an adult male bear in Sweden sometime in the summer. So that was really nice.
Fantastic. Are there many bears where you are around, whereabouts are you? Are you kind of in the south of Sweden?
Well, Uppsala is just north of Stockholm. It's an hour's drive north of Stockholm. So it's very rare to see bears just around here. But if you drive a few hours north, there's quite a few bears, actually.
Interesting. You've spent several years in the field in some 20 countries, in Asia, as well as multiple trips to other parts of the world, including North America and parts of Africa. You are the author of over 100 scientific papers and 60 popular science articles, including many on difficult to identify bird species, which is great. And you're the first author of 2 highly acclaimed books, A Field Guide to the Rare Birds of Britain and Europe, which was published in 1991, and the Pipits and Wagtails of Europe, Asia, and North America in 2006. And you are co-author of chapters in 2 volumes of the Handbook of Birds of the World, as well as a load of other things. And at the moment, you're working on another tome. You're working with my good friend, Dr. Paul Donald, who's at the RSPB. You're currently writing the handbook to the larks of the world, which is supposed to be completed this summer. So number one, have you done your work? And number two, if you haven't, how's it going?
Well, still struggling with it. It's a huge project, takes so much time, and yeah, my main problem is always lack of time. I've got too many tasks, too many projects, so it's hard to finish, but we will finish. The plan is to finish. Plan is to finish before the end of the year. We'll see if we manage that or not. But anyway, it's going to be, we're approaching the end.
Yeah, because the end of the year is only 2 months away. I must remind you, Per.
Oh, shit, just 2 months away. Oh, shit, I thought it was 10 months away. Okay,
what is it?
Yeah, we'll see how it goes.
What is it about larks that interests you? I mean, larks are quite an interesting family of birds. I mean, I remember when we first spoke earlier this year, I was telling you that I went to South Africa and I saw larks and I was thinking, what are you? I was looking right at it. I made sound recordings, video recordings, and to this day I still don't know what they are. What's so interesting about larks?
Well, they're incredibly beautiful. All different shades of grey and brown and buffish, and some streaking, some lack of streaking. I just think they're great birds. And one thing that I've always been interested in is tricky to identify things, and larks really offer some identification challenges. So it's a great group to work with, and also from a taxonomist's point of view, there are surely many more species of larks in the world than are presently recognized. So we're going to see quite a few splits in the future, I'm sure. So it's a really cool group to work on. They've got beautiful songs as well, most of them. So yeah, no, really nice birds.
So currently, how many species of lark are there in the world?
Well, good question. I should know that, but around 100.
100. That's why coming from Britain, I feel so impoverished because we have two species of lark, three. We've got the skylark, woodlark, and then we have the shorelark that turns up every now and again, and that's about it. And I spend a lot of time in Spain, and we've got a few more there, but still not, probably about 6 species, I suppose. I suffer really hard trying to identify Thecla's lark from crested lark. And for the Zoomers who are not familiar, both of them look roughly the same, brown, streaky, with a bit of a crest, but the crested lark's crest is slightly longer. It's got more of a straighter, longer bill, I suppose. No, more slightly curved, isn't it? Sorry, slightly curved bill. Is that right?
It's slightly curved. Yeah,
slightly curved.
Slightly decurved.
Yeah, and it's got a slightly different shape, and I can't. I think I've got it. It's taken me 7 years to get to the point where I can almost confidently say that's a Thecla. But then I made a massive mistake on Facebook recently. No one picked up on it apart from one person who was very discreet. He sent me a message in a private mail as opposed to putting it out in public. I put out a picture which I proclaimed as being a crested lark, and I was totally convinced it's a crested lark. And he wrote to me and said, actually, David, it's a Thecla's lark. So I said, you know what? I'm going to leave it there and see if anyone else spots it. So no one did, so I got away with it. So that's good. Anyway, tonight you're going to talk to us about the general principles of taxonomy, and you're going to show us a presentation about some of the work you've been doing. And please, Zoomers, if you fancy actually asking a question at any point, just let us know, because I certainly will be, because when you speak, Per, I'm always asking questions, and that's not anything against you. It's just that I'm very curious as to some of the things you talk about, because I remember last time you talked about wagtails, and it was really amazing, some of the weird-looking things living in far-flung islands in the Pacific that actually are very closely related to tree pipits and stuff like that. So, yeah, I think you should take it away, Per.
All right, then. shoot. So I'll start by sharing my screen. And excuse me, it's not COVID, but I haven't spoken to anybody for a while. So
I know the feeling.
Can you see that?
Yeah.
And I'll just put it on full screen. So,
by the way, if anyone in the Zoom room can't see this, just send us a sign, send us like a thumbs down or something, so we know we've got an issue. But you should be able to see this. Sorry, there.
Okay, so since this is conservation with you, David, I've got to say something about conservation. So I start by saying that taxonomy is the foundation for conservation. And it's actually, why is this not working? I don't know. Oh, yeah, okay then. And I would say it's the foundation for everything else in the world. And nothing in biology makes sense except in the light of taxonomy and systematics. Well, that's sort of not quite true, but taxonomy really is very important, not the least for conservation, because if you don't know what you've got, if you haven't got names on things, if you don't know how they're related, you don't know what you should preserve, what you should protect. All right, so since many of you probably aren't very accustomed to reading phylogenetic trees, I'll just give you a very brief introduction on how those work, because I know that people are having troubles understanding those. So those three trees that I'm showing here are actually all showing the same relationships, even though they might look different. The trick here is to look at what we call the internodes, so the places where different branches come together. And that shows that B and C are most closely related. They are what we call sister species or sister taxa. Taxa is plural for the word taxon, which means any taxonomic group. That could be a species, a subspecies, or a family or a genus, any kind of group. And then B and C in turn are closely related to no, I can't see, must be A up there. I can't see because there's something hiding the upper part of my screen here. And then D and E again are sister species, and they in turn are sister to this group here. And a group like this is called a clade. If we move on and look at these trees down here, they actually show exactly the same relationships, even though the shapes of the trees are different. If you look at this one down here, you can see that all of the branches are sort of the same lengths here. So this tree doesn't tell you anything about the amount of change that happened along the different branches, doesn't tell you anything about the time or anything. If you look at this tree up here, you can see that the branches have different lengths, which indicates that they are proportional to the amount of change along these branches. And this tree here is a time tree where you've got all the branches ending here at present time. And then you can see that you've got different branch lengths in these trees. So these are all proportional to time, these ones. And here's another way of showing trees. Okay, so remember this. Look at the internodes where branches come together, because that is how you read trees. These are the closest relatives in this tree. This one is the next closest to these, and these three are sister group to these two here. Yeah, so I've already mentioned sister species, and the word clade I've also used. So a group like this is called a clade on a tree. So these are sister clades. Here's one clade, Here's another clade. Here's a third clade. So this clade is the sister clade to this one here. One term that is really important in taxonomy systematics is monophyletic. And that is the same, a monophyletic group is the same as a clade. And that forms the basis in modern classification. So what is a monophyletic group? Well, it's a group that includes all the descendants of a common ancestor. So this group here, for example, has, now I can't see exactly where I'm pointing, but there is a most recent common ancestor just above my arrow there, just above my pointer. And here's another monophyletic group, another clade that includes these three groups here and nothing else. You see they share a most recent common ancestor here, which they don't share with any other group. Here's another group comprising all those, another monophyletic group. Here's yet another one with a most recent common ancestor here that is not shared with any of the others. And another one. So all these are monophyletic groups, and any other combinations, so for example, combining this one with this one here would create a non-monophyletic group. So one important thing here is that this is the family Paridae, the tit family. And this family includes a number of genera. When I was a kid, this family included most of these genus names that you see here, all except this one here and those two, were called Parus. That was the genus Parus, comprising practically all of the tits. But then this bird, it was realized that this bird here, the ground tit, which was previously called a ground pecker and was believed to be a crow of some sort, it was realized that it was a tit. And as you can see here, this one, the ground tit, is actually more closely related to great tits, which is here in this group here, than a great tit to a blow tit, for example, or a willow tit, marsh tit down here. So because of this, because this was never considered to be a tit until it was revealed by genetic and careful anatomical studies, some people felt that we must keep this name, Pseudopodoses. We can't merge this with the tits without, well, feeling bad about it. So therefore, this group here remains as Parus, if we want to keep Pseudopodoses. This group here, which includes the great tit, which is the so-called type species for the genus Parus, remains as Parus. And then, because only monophyletic groups are accepted in modern systematics, this group here must also have a name. And then the name Macrolophus was available. Here's another group that was given the name Melaniparus, and so on, so forth. And as you can see here, the limits of these different genera within this family Paridae differ. So some of them include several species, Macrolophus 8 species, Parus 5, Pseudopodoses a single, Persilla 18 species, etc. So this is actually very subjective. So as I said before, all of these were previously treated as Parus, which would have been perfectly all right. It would have been equally okay to treat these as Parus, these ones here, because they include the type species of Paris. It would not be allowed to call these Paris, because then the type species of Paris would not be included. But you could call all these Paris. That would be perfectly all right. So the take-home message here is that the division of genera, families, and all other higher levels in taxonomy are actually subjective. So it's up to any working taxonomist to say that I want this to have a generic name. I want this to have a generic name. As long as you put these names on monophyletic groups. Okay, so here's an example that I've been involved with. We'll look at the so-called grass birds, the family Locustellidae. So it's grass birds and grasshopper warblers. You've got, in the UK, you've got Savis and common grasshopper warbler. Occasionally river warbler, which is more common in Sweden. But this tree, which we reconstructed based on DNA sequence data, showed that all the ones in red font were placed in the genus Bradipterus. And as you can see, Bradipterus is spread out across this tree. All the ones in black font were placed in the genus Locustella, that is the grasshopper warbler themselves. And then you got Megalurus, this color here, which you've got also in several different places. You've got Syncloramphus, another color. You've got Eremiones, you've got Dromaeocercus, you've got Chanicola. So as you can see here, this is a really weird tree. And I wish to highlight here, you see there's a lot of numbers and some stars on this tree. And they indicate the statistical support for the different clades in this tree. So the higher the number, or a star in this case, indicates that this relationship is very strongly supported by this particular data and this particular analysis. So what we show here, if this tree is correct, it shows that there's a lot of genera that are not monophyletic, and also there's a very, very complex morphological evolution, because these relationships, the traditional classification was based on studies of morphology, so studies of the appearances of the birds, mostly in museum collections. You can note here that you've got one clade here, one group where all of the species occur in Eurasia, another clade, another group where all of the species except one, this one here, occurs in Africa and Madagascar. And this one here, this group here, including this one, which is in a different group, occur in the Orient Australasian region. So what to do in this case? Well, as a taxonomist, I don't like these non-monophyletic groups, and therefore we decided to propose a revised taxonomy of these birds. So as you can see here, all the birds in Eurasia, in this clade here, which form a monophyletic group, all closely related to each other, were placed in the genus Locustella. That includes the common grasshopper warbler, for example. Then this group here, which includes the African and Madagascar birds, were placed in Bradipterus, except for this one here, which we kept the name Chanicola for. You can see here it's more divergent from the others. These are quite close. You see the branch lengths here are quite close, and here's a rather long branch down to this one here, Chanicola. We decided to keep that in a separate genus to retain the genus Chanicola. And here, well, and then these ones here, we placed in the genus Megalurus. And the type species of Megalurus is this one here, Megalurus palustris. And this is not really good, because as you can see, this runs counter to the principles I've just talked about. This is not part of this group. And we decided that maybe this phylogeny, this tree is not actually trustworthy, because we had some conflicting data that suggested that maybe this one is actually more closely related to these. So we said provisionally, we accept this. All right. Some years later, we produced a new phylogenetic tree, including all genera in the world and all except 3 of the species of Locustellidae, of these grass birds and grasshopper warblers. So we added a number of species compared to the previous one. And again, despite that we have revised the phylogeny here, you've got the Locustella here, you've got the Bradipterus here, you can see there are still some oddballs, so things that have different colors. Megalurus, for example, you've got here, 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 here. Bradipterus, you've got here, and you've got up here. And Chenicola, you've got in 2 places. The ones in black font are monospecific genera. So genera that include only a single species, like this Amphilis, Chaetornis, Elaphronis, Breticophorella, Malia. And again, a really complex morphological evolution. Look at this variation. Most of them are just little brown jobs. But then you've got this bright yellow thing here. You've got this big, mostly blackish thing here. You've got really big, long-tailed things. So very complex morphological evolution in this case. I'd just like to highlight again that the relationships here are uncertain. You see, you've got really low support values here, really low statistical support that indicates that this isn't quite trustworthy, not quite robust. So we need to get more data and try again. But anyway, some of the remarkable things here is that the bristled grassbird is the sister species to the broad-tailed grassbird. Both of them occur in India, but the broad-tailed grassbird is not very closely related to the fan-tailed grassbird, which occurs in Africa, despite that they are incredibly similar in appearance. And they were at one point actually treated as the same species. And another thing here is that these birds, which look very similar to each other, are not very closely related to each other, according to our data. And in fact, the striated grassbird is, in our tree, more closely related to these 3 than to these 2. So that's quite bizarre. And based on this, we decided to revise the taxonomy once more. And as you can see here, we used the name Bradipterus for this group here, meaning that Amphilis is a synonym of Bradipterus. So Amphilis had to change name to Bradipterus instead, Bradipterus sibomai. We kept Chenicola, we did not keep Chenicola for this one here, but we kept Chenicola for this group here and synonymized Chaetornis with Chenicola. And in this case, why does Chaetornis get the name Chenicola? Why not the other way around? Well, it's because Chenicola was described before Chaetornis, so it has priority over Chaetornis. So in this case, this group here was called Chenicola, Chenicola platurus, and Chenicola striatus. And for this one, we found a name that we could use, which was Catriscus. So that's Catriscus brevirostris. and so on and so forth. So some taxonomic changes were proposed in this paper.
Is out of interest, to interrupt you for a second,
Yes.
Susie Buttress has just asked, is the uncertainty of species relationships due to a lack of genetic material to analyze, or due to the need to further develop the genetic analysis process?
It could be both, actually. So in this case, we used, I can't remember the number of genes, but we used several different genes, and therefore it's quite possible that with the addition of more genetic material, more longer DNA sequences, we might be able to resolve these relationships. However, it's possible that, for example, you see here really, really short internodes, really short branches. And these short branches indicate that things have happened quickly. And when things happen quickly, when there's like an explosive radiation, it's quite possible that with our methods, we will never be able to really disentangle a sequence of events. So it's quite possible that we will never be able to say for sure what happened first in this case. But I would suspect that with more genetic data, such as whole genome data, we could surely improve this. And see, there were two parts to this question. I forgot the other part. So that was the genetic part. What else did you ask for? Sorry, say that again, please, David.
She was asking, is the uncertainty of species relationship due to a lack of genetic material to analyze, or due to the need to further develop the genetic analysis process?
Yeah, okay, so thank you. So yes, of course. So the methods that we're using today could surely be improved, and with improved methods, we might get better results. We might be able to resolve things that we can't really do today. One problem, as we're getting more and more data, as we're getting genomic data, so millions of genes to analyze, the computer capacity is a huge problem. So we just haven't got fast enough computers to deal with these enormous amounts of data. So that's another problem with looking at those incredibly complex data sets.
Yeah. One other question whilst we're on the subject of these fan-tailed, well, grass birds and what have you. Shailesh, my good friend Shailesh from London, big shout out to you. Actually, I'm not too far from London at the moment, but anyway, what was the old name of the fan-tailed grass bird in Africa? Would you know?
The old English name.
Yeah.
The only no, not broad-tailed grass bird. No, shit. I should know that, but I forgot it.
We can come back to that. We'll come back to that.
It. Yeah. But the scientific name was the same. It's been called Genicola brevirostris for a long time anyway. But it's right. It was not called fan-tailed grass, but it was something different. I forgot. Sorry. Yeah. Okay. Shall I move on?
Yep.
All right then. So these birds, which I'm sure are very familiar to all of you, at least if you're based in Europe, are clearly different species. I mean, they do occur together. We say they're sympatric. They do not interbreed with each other. They differ in multiple aspects. They differ in size, shape, plumage. They've got different calls, different songs, different behaviors, and they're genetically different. And if you remember from the tree that I showed before of the family parody, they are not each other's closest relatives. So these are definitely different species. There's no doubt about it. Nobody would argue something else. But what about these ones here? are these different species. So these are both blue tits, and this fellow here occurs in this area. So it's got it's not sympatric with, it doesn't occur in the same area as this one here. So they differ slightly in structure. Basically, they look very similar. They're much more similar than grey tits and blue tits. But they differ slightly in structure. They differ slightly in plumage. They've got slightly different sounds. The distributions, as we say, are non-overlapping, but there's a deep genetic divergence. So how do we treat these? Well, DNA makes things more tricky in this case, because it's been shown that the blue tits that occur in North Africa and on the Canary Islands, this one here, are very deeply diverged from the ones in Europe, which are more closely related and actually very similar genetically to the azure tit. And azure tit's always been treated as a different species, whereas these two have been treated as conspecific. So in this case, genetics is complicating matters. And as you can see here, there's also rather deep divergence between birds from La Palma and Libya compared to birds from other parts of North Africa and the Canary Islands. So in this case, genetics is really complicating matters. And with respect to the blue tit in Europe and the azure tit, you can see they've got quite a lot of overlap in their distributions, but they also hybridize where they meet. So it's quite possible that the really close relationship that we saw in the tree in the previous slide is actually at least partly due to this hybridization, which might have homogenized their genomes. So more research is needed on this group, but that shows that highlights the difficulty of species delimitation. So what is a species? Well, if you're in the same place as in this case here, this is in Eastern China, it's usually not a problem to define species, because these ones here, which all breed in Eastern China, are clearly different species. They look different, they behave differently, they don't interbreed with each other. There's no doubt about it. But what about these guys here then? The azure-winged magpie in Western Europe and in Eastern Asia, they look very similar, sound similar, behave similarly. They were previously treated as the same species, and it was even suggested that birds might have been transported from Eastern Asia to Europe and released there. That is definitely not the case, as has been shown both by genetics and by DNA. So they really quite deeply diverge these. But anyway, when you have things that occur widely apart, you may have problems. So a species concept tells you what is a species. So it's the ideas of what a species are, the concept of species. Species criteria, on the other hand, describe how different taxa, so how different groups of organisms may be delimited. So, see, I can't read because there's something on top of my screen here, but yeah, it should say species sensu Charles Darwin up there. Yeah, so Charles Darwin, he wrote, in determining whether a form should be ranked as a species or a variety, the opinion of naturalists having sound judgment and wide experience seems the only guide to follow. And I look at the term species as one arbitrarily given for the sake of convenience to a set of individuals closely resembling each other. So that was Charles Darwin's view on species. And I looked at the Encyclopedia Britannica and the Free Dictionary to see how they define species. And the former, the Encyclopedia Britannica, says that species are related organisms that share common characteristics and are capable of interbreeding. And the Free Dictionary says a group of closely related organisms that are very similar to each other and are usually capable of interbreeding and producing fertile offspring. So this is a bit odd, isn't it? So they're usually capable of interbreeding and producing fertile offspring. That means things that can be the same species are not necessarily producing fertile offspring. Anyway, a comment on this is that many species are capable of interbreeding, and many of those actually produce fertile hybrids. But this is important. Different species usually don't mate with each other. So the figure down here showing ducks shows that the pintail can produce fertile hybrids with several other species of ducks that look totally different from each other. But importantly, normally, a female pintail would not mate with a male mallard or a male gadwall or a male teal or any other male duck except a male pintail. So that's the important thing. Different species usually don't mate with each other. Even if they do, they could produce fertile hybrids in many cases. So the biological species concept was formulated by the late Ernst Mayr, who said that species are actually or potentially interbreeding populations, which are reproductively isolated from others. So interbreeding and reproductively isolated, those are the two main things of the biological species concept. Joel Craycraft from the American Museum of Natural History has formulated a phylogenetic species concept which has gained some popularity in ornithology. And according to his definition, species are the smallest group that are diagnosably different from others. So diagnosably different means they differ by some features. It could be a color pattern, it could be a size, diagnosably different size difference. It could be some other morphological feature. It could also be some genetic features. And there are many, many more different species concepts. I think there are at least 60 different species concepts proposed. So what are the differences between these? Well, the BSC, the biological species concept, is actually a prospective species concept because it focuses on interbreeding and reproductive isolation. What is important here is that you're looking at what might happen in the future. Will they come together? Will they produce a lot of intermediates? Will they actually fuse sometime in the future? Or will they stay separate? There are usually no special criteria for delimiting those groups that you should treat as either species or subspecies. So subspecies are usually recognized under this biological species concept. And there's no generally accepted definition of subspecies, but you could say that a subspecies is a geographical variant that is not reproductively isolated from other subspecies that are classified as the same species. And the biological species concept accepts non-monophyletic polytypic species. So that means you can have a species that is polytypic, which means it has multiple subspecies, but some of these can be, these are not, they don't form a monophyletic group, these ones. The phylogenetic species concept, we'll look at this in detail. So the phylogenetic species concept is retrospective in the sense that the evolutionary history is important. It's more important what has happened than what might happen in the future. So we have species, we can observe them today. We don't care about what might happen sometime in the distant future. And the smallest name-bearing units, the ones that are called species in this case, are defined by those diagnosable characters. And they usually don't recognize subspecies. And they don't accept species that are not monophyletic. So criticism here. The evolutionary history may be misrepresented under the biological species concept. If we have 3 different species here, A, B1, B2, this is the relationship between them. So as you now know, these 2 are more closely related to each other than any of these to that one. So this is the sister species to these 2, according to this tree. If A and B1 are reproductively isolated, they don't interbreed with each other, or if they do, they don't form any fertile offspring. Same with these 2, A and B2 are not reproductively isolated, but B1 and B2, they actually interbreed. So they interbreed and they may form fertile hybrids. They are treated as the same species under this biological species concept. Another problem that has been highlighted is that it's hard to say whether things that occur in different areas, that's called allopatric, whether they would interbreed and to what extent if they would come into contact, because they're isolated, so we can't know that. And also, as you know, there are hybrid zones between different species, and there are various degrees of interbreeding in those hybrid zones. Some birds, they interbreed a lot in those hybrid zones, others just interbreed a little bit. So where do you draw the line? That's sort of tricky. And it's also been shown that complete reproductive isolation is very slowly evolving in birds. And also, there aren't any asexual birds, but that's another problem with this species concept. So the phylogenetic species concept then, diagnosability. So how you define your species, the characters that you use can be really subjective. I mean, you can imagine that you can go really, really deep down and look at minute differences. So how tiny differences are okay for delimiting different species. Okay, so that's what I said there. And what proportion of those individuals that you're studying need to be diagnosable? Is it okay if only 75% can be identifiable, or do you need all of them to be? Is it okay if just one of the sexes can be can be identifiable? But importantly, for both these concepts, there are very often problems for things that occur in close contact or in different areas. So things that have abutting distributions or that are completely separated from each other. And especially with things that have evolved recently. So recently diverged organisms, groups of organisms, that's the main problem. And that problem is the same for both these species concepts. So to conclude, all species concepts, in fact, are problematic. So why is that? Well, let's look at speciation, how that usually proceeds in birds. So imagine we have a single species here represented by this blue blob. And you might have a separation into 2 parts. like this. It could be, for example, a rift, like the Rift Valley in Africa. It could be expansion of deserts. It could be expansion of a sea area, an oceanic area, which has happened multiple times during glacial periods. Sea levels have risen and sunk down again, so islands are being created. It could also be, well, an island where something is expanding to. So a population might migrate to this little island. So with time, differences in these isolated populations will surely evolve. And they will evolve slowly and gradually. Sometimes they might evolve faster, sometimes slower. So there's no constant rate of change, at least not in appearances. With time, maybe this population that has been isolated might come back into contact. That's called secondary contact. And maybe in this case, the differences that have evolved in isolation might be sufficiently large to make these populations reproductively isolated. So maybe they don't mate with each other anymore. If they mate, maybe the offspring, the hybrids, are not fertile or have poor survival rates. So reproductive isolating barriers could be, for example, sexual signals. They don't mate with each other because they have different songs, they have different displays. There could be sexual incompatibilities. That's not so common in birds, but it's common in many other organisms. There could be ecological differences. They might occur in different habitats. There could be differences in timing of breeding. So maybe they breed at different times of the year, so they don't actually interbreed for that reason. There could also be genetic incompatibilities that prevent them from producing fertile offspring. So why then is there a problem to delimit species? Well, I would say it's innate to the evolutionary process, because speciation is a gradual process, and a gradual process creates a continuum through time, and such a continuum can only be subjectively divided. So if we look at this species here, we say here's species A, and with time this has split here into two different groups, two different populations that don't have any contact with each other. So with time, they will diverge. And are they sufficiently different to be treated as different species at this point in time? Or maybe not until this point in time? Or perhaps not until here? So hopefully you agree with me that it's really hard to draw the line between when are these sufficiently different to be treated as different species. And you could think of this, you could compare this to, for example, age, height. So a young person versus an old person, it's a gradual thing. You at one point in life you're young, and then you gradually become older. But when are you old? And another important thing is that the rate of change in different groups of organisms, in different populations, and in different traits such as plumage, structure, vocalizations, etc., are not changing at an equal rate. So the rate of change is highly variable. Think of the larks, for example. Many of the larks many of the warblers, they look very, very similar to each other, even though they might have been separated for millions of years. Whereas the yellow wagtails, for example, the different forms of yellow wagtails that we usually refer to as different subspecies are very, very young, but they look very different from each other. In other cases, you might have a very fast divergence in songs, for example. In other cases, vocalizations don't change very much despite long time periods. So the rate of change is very unequal in different lineages, different groups, different populations. So here is a theoretical suggestion for a solution to the so-called species problem. And this was formulated by Kevin de Queiroz in a series of papers. If you look at a species here, with time, after a splitting event, after a separation into two different groups here, you see they become gradually different. And at this point in time, which is today, they really are very different in many, many different aspects. And so what he proposed is that different species definitions are actually just variations of the same general species concept, which he calls the general lineage, the general concept of species, or the general lineage species, the general lineage concept, yes. And so, for example, diagnosable characters might have evolved already down here, but these two might be reproductively compatible, so they still could interbreed with each other. But then these are different species according to the phylogenetic species concept, but maybe they're not different species until this point in time when they become reproductively isolated. So therefore, you could see this as different species concepts are focusing on different time periods in the speciation process. Okay. Now that we know exactly what species are, we'll look at the number of species in the world. So Sharp, he was working at the so-called British Museum of Natural History in those days, now the Natural History Museum. He identified 18,900 species in the world. Whoops, sorry, pressed the wrong. And then came Ernst Mayr, who formulated the so-called biological species concept. And he thought there were too many species in the world, so he lumped many of the different he lumped many of the species into the same species and recognized the different groups as different subspecies. So you recognize 8,590 species. Oh, wrong way again. Then came Peters some time later, number gone up a bit. Clements, even higher number of species. Dickinson Remsen, over 10,000 species in 2013, 2014. Del Hoyo and Koller, 2014, 2016, 10,964. So as you can see here, the number has increased quite dramatically since 1951. Over almost 2,500 species have been gained in that period. But there could be even more, because Barraclough et al. Suggested in a paper that there could be, now I can't read this, but it's over some 18,000 species, according to them, might be valid. And their estimate is based on comparisons of different, mainly genetic studies that show that there's a lot of subdivision within currently recognized species. Okay, so why is this incredible increase in the number of species? It's not mainly because of newly described species, because between 1950 and 2016, only approximately 300 new species to science were described. And in the same period, some 30 to 40 species went extinct. So this increase, this strong increase is very apparent in especially some groups of birds, like the reed warblers. If you look at Peter's checklist from 1931 to, what does it say, 19? I can't read here. I hope you can see the full screen.
1987.
Thank you, 87. And compare that to Gillendomsker in 2019, you can see within the reed warbler family, there's been a 50% increase. And within the leaf warblers, there's been also 50% increase. Same with the albatrosses, larks, slightly less, but over 30%, gulls, some 20%, alcids, and ducks. And the question is, next time we evaluate this, how many species will there be? There will surely be more species in all of these groups. And my prediction is that the larks is, of these groups at least, the one that will have the largest increase in the number of recognized species. We'll see if it tops 130 in a few years. So I give an example based on research that I've been involved with, the Lesser Asian Sandlark complex, Lesser Short-toed, Asian Short-toed and Sandlark complex. That's how it should be called. And this is based on two different papers. The latest paper was accepted for publication just a few days ago, actually. So it will be out online very soon. It will actually be open access, so anybody can read it, even if you're not affiliated with any university. By the way, when it comes to scientific papers, in case you want any that you can't access, you can contact me and I can help you. So these larks are usually treated as 2 or 3 species. So most checklists recognize the sunlark here as one species. It's the yellowish coloration here. So it's the southernmost of these birds. And the lesser short-toed lark is sometimes considered to have this huge distribution. Sometimes it's split into 2 species. In the western part is the lesser short-toed, and in the eastern part is Asian short-toed. But there's a lot of variation among different authors here. So some authors think that, some authors, well, there's no, they don't always treat the same taxa as the same species. So there's no, lost the word. Anyway, there's differences among authors. So what we did here is that we traveled quite a bit and we collected DNA samples and sound recordings and morphological data, so plumage and structural data. And of course, this is not only done in the field, also a lot of museum visits. And as you can see in this picture here, they are pretty similar. Can you spot any differences? So this is Chilensis, Asian short-toed lark, as it's sometimes called, and this is Heine, which is usually called lesser short-toed lark. These birds were all collected very close to each other in Mongolia. Really, really similar looking birds. However, they usually differ in tail pattern and in wing formula. And also you can see slight differences in wing coloration, in pigmentation. So these birds have got darker pigmentation, which also make the feathers more resistant to wear and abrasion. So you can see these feathers here. These photos were taken at the same time but these feathers are considerably more worn than these ones, only because of the less dark pigmentation. So dark pigmentation protect feathers. Whoops. Right, so there are at least on average differences in these respects between these groups. And when you look at morphometrical data, this is a figure which Paul Donald has done. I don't know whether Paul is listening to this or not, but this is from our paper, which Paul was also co-author of. And so he has just a lot of different measurements, and all the ones that have the same color are not significantly different from each other, whereas the ones in different colors are significantly different. So you can see in some respects, you've got similarities, and in other respects, you've got differences between different groups of these larks. And when it comes to songs, you could identify at least 4 different groups, and we'll see if I can play the top one here, Hey Nay. You see the sonogram here, and I'll see if this works. So the song, as you can see here, is very drawn out, a very long song, and it usually begins rather stuttering, rather sort of slowly, and then speeds up. And the next one I will play is this one here, Chilensis. So a shorter song, which begins much more abruptly. So there are differences among all those. I'm not going to play the others, but there are differences among all those. And of course, there's a lot of individual variation, but we've studied lots of individuals, and we've concluded that they differ in multiple aspects. And here is a plot showing differences between these groups. So this group here, which we call Hey Nay, has one type of song, which is here in this plot, and the rufescens group, which is the westernmost group from Europe to Western Asia, is in another part of this plot. And then you've got like a continuum from within the Eastern Asia, from Western China all the way to Eastern China, Eastern Mongolia, and you've got the sunlark up here as well. So at least 3 different clusters. When you also look at sonograms and listen to these birds, I would say that these ones are actually quite more distinctive from these birds than the look here in this plot.
Before you carry on, sorry, Per, can I just interrupt for a second? We've actually gone over the hour. We're an hour and 10 minutes. So what we're going to do, we're going to officially end it now. So it means that if you're watching this in the future, I feel like back to the future now. If you're watching this in the future, you're going to have to become a member of the Urban Birder Club to see the rest of this. Okay. And I just want to, at this point, thank you, even though we're going to carry on into the party zone. But thank you very much, Per, for actually being here tonight to give us this fascinating talk. There's lots of questions here, which will actually be answered shortly, hopefully, when we ask you the questions. But I just want to say at this point that the upcoming In Conservation With will include tomorrow night Kate Maguire, who is an artist who uses the feathers of pigeons predominantly, but other birds to make art. She's not really a birder as such, but she's really quite an interesting woman, and she's got quite a lot of stuff to say about pigeon fancying as well. So that could be quite a contentious conversation tomorrow. And on Thursday, I've got 2 young men who set up this whole thing called Flock Together, which is all about getting ethnically diverse people out birding in London. And this whole thing's kicked off now. It's become nationwide. Sorry, not even nationwide, worldwide. I think there's a branch in America now. So it's actually spreading across the world. So we'll be talking to them on Thursday. And then there's a few more people coming up over the coming weeks. But I will end it there in terms of what's happening in the next couple of days. So I'd love to see you guys again for the upcoming in conservation with. Thank you very much again